Welcome everybody to this afternoon's session. If you're here, you are here to listen to Dr. Joanna Klubo, where topic is a presentation on clinical trials, what they are, and all the way to trials at the National Institutes of Health. Dr. Klubo is a Lasker tenure track investigator and acting section chief of thyroid tumors and functional thyroid disorders at the National Institute of Diabetes and Digestive and Kidney Diseases, National Institute of Health in Bethesda, Maryland. She obtained her MD and PhDs from Nicholas Copernicus University, Collegium Medicum in Poland, and Master of Health Science degree from Duke University in Durham, North Carolina. She underwent residency training at Nicholas Copernicus University, as well as Georgetown Washington Hospital Center in Washington, D.C., and a fellowship in endocrinology at the NIH. She is board certified in internal medicine and endocrinology, diabetes, and metabolic diseases. Dr. Klubo's primary scientific focus involves translational and clinical studies focused on the optimization of thyroid cancer care using personalized medicine. Dr. Klubo is a recipient of several awards for her work, including American Cancer Society for Clinical Investigation 2021 Young Physician Scientist Award and Women Advancing Thyroid Research Award from the American Thyroid Association. Dr. Klubo is a 2011 ThyCa Research Grant recipient. I turn you all over to Dr. Klubo.
Thank you so much for this kind introduction. So yes, it is a great pleasure to start giving back to ThyCa. Yes, indeed, I was an ATA grant recipient several years ago and continue to work to optimize care for patients with thyroid cancer. I wish I was there in person. I love interactions with patients. However, government has its own rules. It's end of government fiscal year, so just this travel was not possible. That said, so clinical trials, back to basics. Let's start with the bigger picture and the purpose of clinical trials. So obviously, we are interested in clinical trials devoted to treatment, but also there are clinical trials that help with establishing diagnosis, detection, prevention of certain disorders. And as a member of the NIH community, we particularly focus on translational research. So bringing the question from the patient's bed, trying to investigate the pathophysiology behind certain disorders in the lab and preclinical studies, and trying to translate it to first in human clinical trials or more advanced clinical trials that sometimes provide surprising results, and therefore, in order to understand the outcomes of the clinical trials, we oftentimes go back to the patient's bedside, trying to investigate what was the reason for unexpected outcome. So what are the key components of this bench-to-bedside translation journey? First of all, it is important to identify relevant clinical question, formulate appropriate hypothesis, plan experiments to test this hypothesis, do it in preclinical models until everything is set and we are confident that we can move to this big step of translating our findings into clinical trials. And how does it occur? Obviously, it is very tightly regulated by Food and Drug Administration. So if we plan to apply anything novel, not standard care, into the clinical trial, we need to apply for investigational new drug process, so-called IND. And this IND, in order to be granted, needs to be based on robust animal study data and toxicity data to even consider starting a phase 1 clinical trial, manufacturing information, making sure that all the requirements of processing and manufacturing of certain agents are met. These are very tightly regulated as well. Study plans need to be very detailed and analyzed by scientific community and regulatory community. If there are any data from prior human research, for example, if we are translating, let's say, a medication from one disease to another, we also need to rely on what's out there, what's available. And obviously information about the investigator, do they have what it takes to conduct a clinical trial? So what's the purpose? Yes, it is to document that certain agent proves to be safe and effective in regards to therapy or diagnostics. And patients come first. The concept of clinical trial comes from some deficiencies, need that we do see in the patient's bedside. But once the concept is established, in order to start clinical trial, there is multiple steps to be undertaken in order to make it happen, including all the pretrial preparation and management, regulatory compliance, and so on and so forth. And as I mentioned, we are back to basics, so I would like to just reiterate what certain phases of clinical trials entail. So if we are talking about bench to bedside translation, obviously the first step is a phase 1 clinical trial. These trials usually are the smallest. They include from 15 to 90, 100 patients. Usually 70% of the drugs that are tested in this phase can move further down. And I'm going to describe what type of questions each phase addresses later on. But let's now focus. So if we have successful phase 1 clinical trial, we can move to phase 2 clinical trial, which is slightly larger. And again, in regards to what is the efficiency of translating the drugs from bench to bedside. Here, 33% of the drugs move to the next phase. Phase 3 clinical trials is something that has been discussed robustly during this meeting. Dr. Alan Ho mentioned all these big tyrosine kinase trials, decision trial, for example. So these do contain several hundreds, even several thousands of patients, dependent on your primary objective. And usually, if they are conducted, 25% of the tested drug move to the next phase. And phase 4 is already analyzing approved by the FDA drugs, so they are already on the market, but they are in clinical practice, but we learn some additional things about the drug. So let me walk you through this process step by step. So again, phase 1 clinical trials addresses the question, is the investigated agent safe? So safety is the main purpose of these trials. They could be first in human trials or trials on escalating the dose of certain drugs, bioavailability, drug-drug interaction, or food effects on the drugs. And this is how we understood phase 1 clinical trials for many, many years. Right now we are shifting a little bit, and I will tell you why. Excuse me, was there a question? No, this was okay. So how the classical concept looks like, we enter 3 patients into the first dosing regimen of the certain drug, and we look for a safety. So is there any adverse event after this certain dose of the drug? in these 3 first patients. If no toxic response, we can consider escalating the dose. If 1 toxic response, we need to add more patients to the same dose to see if this was just by chance occurrence or there is something to it causing toxicity. And if majority of patients, 2 out of 3, present with toxic response, you know, the drug is too toxic. And we go through this route step by step. Usually at least 9 patients are included. And the main question addressed here is, what is the maximum dose that is associated with acceptable toxicity? However, as you can immediately ask, is maximum dose always optimal? What about efficacy of such drug? Why are we asking only the question about toxicity, particularly in trials in oncology? So, and these questions have been asked, and particularly in oncology, when, as you hear from a couple of talks today, we tend to be more utilizing more personalized approach, targeting certain types of mutations. So therefore, what has been noticed is that current clinical trials, when we have information about genomic markers, leads to responses, meaning partial responses, complete responses, even up to 40% of patients. So there is some efficacy in phase 1 clinical trials that prompted questioning if this, 3+3 design that I just alluded to, is it optimal? And therefore, nowadays, what we try to do is to combine phase 1 with phase 2 clinical trials and ask a question that is the most important clinical question. With every single medical intervention, it's always benefit versus risk. So why don't we ask this question up front? Again, efficacy to toxicity trade-off is what modern clinical trial designs tend to offer. So again, we start with the first certain dose that we think could be safe and effective, and we collect data on both efficacy and toxicity. And this is designed in a way that mathematical model, Bayesian modeling, asks this question with each patient. What was the response? What was the toxicity? And based on each patient that is enrolled in such trial, we can say, okay, are all doses overly toxic and ineffective? If yes, we terminate early. We are not moving forward with such trial. Do we have a reasonable risk to benefit ratio? We move forward, we increase the dose, we enroll more patients. Based on preset objectives of the study, have we reached the number of patients that would let us tell with confidence, this is sample size, this is the number of patients that meet our risk to benefit trade-off, this is when we can stop the trial. And as you can see here, we are not asking a question about maximum safe dose. We are asking a question about optimal dose that is efficacious at the same time with limited toxicity. So this is where the field is moving forward. But let's also discuss phase 2 clinical trials. Again, these are not as large as phase 3. They tend to ask therapeutic exploratory question, help understanding drug disease effect, provide additional safety evaluation, and also maybe optimization of the dose for those bigger phase 3 clinical trials. And they could be divided into proof of concept ones, so we determine the drug's mechanism of action, or dose response studies to determine optimal dose to use in further studies. There is another group of clinical trials that is more commonly used nowadays. Again, we are bombarded by omics, genomics, proteomics, transcriptomics, and the knowledge gained from characterization of different cancer types led to putting them together based on mutation drivers, based on these oncogenes that tell the certain cancer cells to grow, to proliferate, to metastasize. So basket trials, what is constant is the same mutation driver, but they could include different cancers, colon cancer, melanoma, thyroid cancer, driven by BRAF, for example. So this is a relatively new concept, and there are several trials such as these, trying to target molecular pathways, oncogene-driven signaling pathways that are typical for this particular mutation in different cancer types. And the clinical trials that you are probably interested the most, phase 3 clinical trials. So here, what is the question that is being asked? Is this particular agent better than what's currently available? So usually, in order to address such question with confidence, we need to have a large patients population. Therefore, those trials tend to be multi-center, often international, including hundreds to thousands of patients. We are comparing efficacy with established standard care, and this is the most common scenario for patients with cancer, but for other disorders, it could be also placebo. Study participants are usually assigned to groups randomly, and moreover, neither the patient nor the doctor knows what type of treatment are they on, what we call double blinding, in order to avoid any bias in primary objective of the study. So phase 3 are more focused on efficacy than safety. And once phase 3 clinical trial proves to be successful, meaning that we do have benefit that is predefined as compared with standard of care, this is the time that we can file new drug application to the FDA, so-called NDA. And again, this process is lengthy, tedious, and associated with many regulatory steps. There is another type of clinical trials that I also mentioned before, the largest one, phase 4, meaning basically that we are continuing to capture the data of already approved agent in order to address the question, what else can we learn from this treatment or diagnostic procedure? So here we are able to address a question of very long-term side effects or very rare side effects. We could assess quality of life. We could determine cost effectiveness of certain therapeutic or diagnostic approach, and try to identify optimal use, which groups of patients benefit from certain treatments best, which patients are particularly at risk of certain side effects. So these are easier to address with phase 4 clinical trials that are longer term follow-up observations. So this is an overview of how clinical trials are constructed for you to know what you may expect from certain clinical trials that are offered to you. But if you were to search for clinical trial yourself, there is a website, clinicaltrials.gov, that includes clinical trials that are registered. These are international clinical trials, so with sites in the United States. And you just type in your condition, your disease, thyroid cancer as an example, and you could take a look at all studies conducted for this particular disorder, or you could focus on recruiting or not yet recruiting studies. You could also narrow down your interest to certain geographical areas. But if you just, I just did it this morning, if you just type in thyroid cancer, recruiting or not yet recruiting, what comes out is 990 studies that are focused on different aspects of management of thyroid cancer. So with this background, I would like to draw your attention to clinical trials that are going on at the National Institutes of Health, our institutions. As you may know, this is the whole center is devoted to clinical research. Building 10 on this picture is the one where both inpatient and outpatient clinical visits occur. However, campus is much bigger and there are many laboratories that do preclinical work in order to make these trials happen. In regards to NIH, it supports a lot of research endeavors. In fact, 171 scientists either at NIH or whose research has been supported by the NIH funds have been the sole or shared recipients of 102 Nobel Prizes. And in regards to staff scientists within the NIH, last Nobel Prize was received by Dr. Harvey Alter on his work on hepatitis C a couple of years ago. So given this structure, many labs, so clinical centers surrounded by many labs, the most common type of research going on at the NIH is this bench to bedside translation that starts, as I mentioned, with identifying relevant clinical question. So let me walk you through this bench to bedside translation that currently our group is trying to implement. So our question was, can we identify a new therapeutic approach for oncocytic thyroid cancer? And why is that? Oncocytic thyroid cancer is an orphan disease, it's a rare cancer characterized by limited therapeutic option. It is characterized by unresponsiveness to standard therapy, particularly to therapy with radioactive iodine. Only 30% of patients have sufficient uptake of radioactive iodine in this cohort. So question is impactful, potentially impactful and significant. So we move forward with this question. A side note, oftentimes there is some serendipity in science. And we were driven in terms of testing certain new molecular targets for oncocytic thyroid cancer by our other study. This was the study using so-called gallium dotatate. So this is like PET scan, positron emission tomography scan that utilizes an agent called gallium dotatate. This agent targets a certain molecules called somatostatin receptors, and they just lighten up on imaging of the whole body. And what we found is that they tend to lighten up in the thyroid from time to time. And we had no idea what the significance of it could be. So here you've got thyroid nodule. This is the same nodule visualized on the ultrasound. And there is a clearly uptake of this, focal uptake of this dotatate in this particular region. This agent is utilized routinely and FDA approved for imaging of patients with neuroendocrine tumors. And this was a study that just analyzed imaging studies in 237 patients with neuroendocrine tumors. And we found that one out of 10 had uptake in the thyroid, and 1 out of 5 who had positive uptake over there had thyroid cancer. So we asked the question, is it something that could be targeted both with imaging and therapeutically? How often do we see it in thyroid cancer? Is there any particular type of thyroid cancer that is characterized by presence of these somatostatin receptors? So the concept is simple. So we hypothesized that on tumor cells, in this case, thyroid cancer cells, there is receptor, in this case, somatostatin receptor, that could be targeted with this dotatate, and if that could bind to the receptor. So it just goes and sits on the cell surface. And if we attached to this dotatate certain isotopes, certain radiation emitters, we could use it for different purposes. And this is what we call theranostics. So it comes from 2 words, therapeutics and diagnostics. So if we attach to this dotatate gallium or copper, we can utilize it for imaging using PET-CT, positron emission tomography. If we attach lutetium or yttrium, we could utilize it for treatment. And there's also a very active research going on alpha emitters that have very high energy in theranostics world. So then question becomes, okay, we observed this uptake in thyroid, and 21% of tumors were thyroid cancer. How often these somatostatin receptors are expressed in thyroid cancer? Do those cells have it routinely, or what is about this phenomenon? And we hypothesized that it can serve as a molecular target for imaging and treatment. And we were asking a question, could it be in oncocytic thyroid cancer for which we have limited therapeutic options? So when you formulate a hypothesis, you try to utilize whatever publicly available resources are there. So we went to Protein Atlas and found that yes, somatostatin receptors, which I will refer to as SSTR2 over here, are expressed in thyroid cancer, approximately 50%. So this is when they analyze tissues of thyroid cancer and stain them for presence of these particular receptors. We also did it ourselves. So we utilized tissues derived post-surgery from patients who were operated for neuroendocrine tumors. So it served as our reference, as our positive control, and different types of thyroid cancer. And this brown staining that you see over here reflects presence of somatostatin receptors. And these examples are examples of slides with high expression. So it translates to this score 3, that the majority of cells had expression. But we quantified it and found that actually this high expression was found in all oncocytic thyroid cancer patients, but we had only 4 patients. We moved further with this question. We also screened the patients who we knew metastasized, so whose thyroid cancer spread outside of the thyroid to the lymph nodes, to the lungs, to the bones, and imaged them with this gallium dotatate. So we performed PET-CT. You can see all these yellow spots are cancer cells lightening up. So here in the bones, in the lungs, and mainly in the bones in this patient. So bottom line, we quantified this uptake and found again that it was observed particularly prevalently in patients with oncocytic thyroid cancer. But again, we had only 5 patients here. So we continued this clinical trial using dotatate PET-CT in patients with metastatic thyroid cancer, and we continue doing so. So if there is anyone interested in the room, particularly patients with oncocytic thyroid cancer, in receiving imaging from head to toe, utilizing different modalities, including this dotatate, This is the information. We highly encourage you to check this out. We have completed recruitment for patients with differentiated thyroid cancer and medullary thyroid cancer, but we still have this protocol open for oncocytic or HER2 cell thyroid cancer. So, okay, we see that there is a signal here that a certain group of patients might be characterized by high uptake of dotatate. So we know that it could be used diagnostically. How about therapeutically? Could we target these somatostatin receptors or SSTR2 for therapeutic purposes? So again, bench to bedside translation, one of the requirements is to have robust preclinical data, including animal models. So how does do those models look like? So we have established a human cancer cell line, and we just transplanted it to the mice, and the mice do grow a tumor. And we tested different analogs of this SSTR2s, revealing that there was particular one called lutetium dotaebitate that resulted in significant reduction of growth of these tumors in the mice. Obviously, you need to be confident, you need to repeat your experiments, and here we repeat it in the larger group. And again, this lutetium dotaebitate resulted in significant reduction in tumor growth with good tolerability. The mice were not getting sick. So, summarizing this part, treatment with lutetium dotaebitate extended survival and reduced tumor size in mouse model with tumor somatostatin receptor analog uptake that was comparable with the one that we observed in patients with oncocytic thyroid cancer. We quantified it, there is so-called standard uptake values, and this was comparable. So we decided to translate these findings into a clinical trial. And before we do so, we try to collect data from whatever is available, other human trials. So there were ones in patients, again, with neuroendocrine tumors, not with thyroid cancer, showing that this ebitate has 8 times higher tumor dose delivery, so higher accumulation in the tumor as compared with dotatate. However, this is due to this EB moiety that you can see here, that it just stays longer in circulation. So if you have higher uptake in the tumor, you ask the question, how about my normal organs? Am I at risk of side effects? And indeed, dosimetry studies revealed that you can have 3 times higher kidney exposure and bone marrow exposure after ebitate as compared with dotatate. So then the question becomes, okay, so I cannot go empirically with the dose. I cannot go blindly. I need to tailor my dosing towards particular patient's clinical situation, patient's body habitus, clearance, function of the kidneys, liver. Moreover, we know that in order to destroy the tumor, it's not only about the dose, but also the tumor characteristics. How big it is, how fast does it grow, what's the molecular landscape, what's the tumor microenvironment. We heard a lot of information about immune landscape. And what dose actually is going to get in? What is our predicted dose that could potentially destroy the tumor? So in order to address this, we decided to go with, to propose so-called dosimetry. So we are going to individualize in this phase 1/2 clinical trial, we are going to individualize the dose for each patient. So what are we doing? We are delivering just tiny diagnostic dose and check how is it distributed. So we obtain blood samples at 4 time points to assess beta radiation. We performed 3D SPECT images at the same time points. And based on this, we calculate the dose that would not exceed certain thresholds for the kidneys and for the bone marrows. to assure safety. And we do it for each patient individually each time. So currently this trial is under IRB review. We have received study may proceed letter from the FDA. So hopefully within the next couple of months, it will be all open. And what we aim to demonstrate that dosimetry is feasible approach to this individualized peptide receptor radionuclide therapy. We would like to establish safety and efficacy profile of this lutetium dotaemitate. We would like to evaluate association between absorbed dose in normal organs, which we'll be able to quantify with this dosimetry, and safety profile. So is there any threshold that results in, for example, elevation in liver enzyme? We would like to establish the optimal dose. Again, we said about this trade-off, benefits to risk, right? Based on balance of safety and efficacy data. So we do utilize those modern approach to phase 1/2 clinical trials. And then the bigger question and very important one is there any threshold? How much radiation do we need to deliver to the tumor to be able to eradicate it? So we need to associate efficacy with the tumor dose. So this is the plan for this study. And that hopefully will be open within the next couple of months. And we do believe in personalized medicine and individualized approach. So let me share with you also some other phase 2 clinical trials that try to accomplish this goal, individualized approach. In this case, to standard treatment. So we treat patients with radioactive iodine, but we try to implement it in individualized fashion using whole body and lesional dosimetry. So patients with thyroid cancer that spread outside of the thyroid gland to the lymph nodes, to the lungs, to the bones, are subjected, they are candidates for radioactive iodine therapy that could be delivered either empirically, just one size fits all, 150 to 200 millicuries dose, or could be delivered via dosimetry, utilizing the procedure I described. We also recognize the growing population of patients who have radioactive iodine non-responsive thyroid cancer. So we also invite such patients. They are undergoing redifferentiation therapy. As in this example, this is the patient with BRAF mutant thyroid cancer who had no uptake over the lungs. These are the lungs, completely no uptake. 3 months of the dabrafenib resulted in significant induction of the uptake, and it was also confirmed in post-therapy scan. So metastatic thyroid cancer, radioactive iodine avid and non-avid after redifferentiation. These are the candidates to be treated with individualized dose of radioactive iodine. We administer this radioactive iodine under TSH stimulation. So you need to stimulate the radioactive iodine uptake. This is a standard procedure, either by stopping thyroid hormone or by performing injection of recombinant human thyroid hormone. And also we need to make cancer cells hungry for iodine. So low iodine diet is recommended for 2 weeks before radioactive iodine treatment in this protocol. And again, similarly to what I have described previously, what does this study entail? The patient is administered an oral dose of I-124. So this is radioactive iodine isotope used for PET imaging. And we do collect blood and perform PET-CT images. And at this time points, all the values that we do obtain enable us to say what's the maximum dose that would not hurt bone marrow and how much radiation we predict will be accumulated in the tumor. We are also offering this longitudinal phase 4 clinical trial. So here we are addressing a question, what can we learn from application of standard of care? We target patients with either benign or malignant thyroid nodules that are being followed at the NIH. And in fact, through different principal investigators, this study started in 1977, so we can also capture the different therapeutic approaches over time and what we've learned. For example, the big difference is change to the extent of surgical approach, change in indications for radioactive iodine treatment. So we do learn over time. We try to optimize therapy of thyroid cancer patients over time. So for this protocol, who is eligible? Patients with thyroid nodules that require biopsy, patients with recent diagnosis of thyroid cancer requiring consultation, counseling about therapeutic options, patients with established thyroid cancer requiring specialized studies such as radioactive iodine-based dosimetry. And we also enroll higher risk patients with radioactive iodine non-avid disease to screen them for eligibility to be enrolled in other protocols. There was also a session, I believe, 1 hour ago on familial non-medullary thyroid cancer. We also have a longitudinal protocol covering this cohort of patients. This started in 2010 by Dr. Bebu, and it's been continued by our group. So who is eligible? Families with 2 or more members affected by thyroid cancer. Usually the estimated prevalence of such clinical scenario is between 3 to 9% of all thyroid cancer. There are conflicting data on the aggressiveness of familial disease as opposed to sporadic, because we know very little, as indicated in the talk previously, on susceptibility genes. We do not have clear-cut guidance on survival programs, so they need to be optimized. So we do invite and recruit patients with familial non-medullary thyroid cancer, and I'm more than happy to share this information, share this slide with ThyCa if this is what will be of interest. What is the goal here? To evaluate natural history of patients with familial non-medullary thyroid cancer, both affected and unaffected family members. Identify the best surveillance strategy and identify susceptibility genes, as discussed again with our genetic counselor in previous sessions. It might be that there are many genes involved, not just one culprit gene in this disease. What have we learned from this trial so far? So we did perform screening with ultrasound of affected and unaffected family members. And in regards to unaffected family members at baseline, if they were coming from family when 2 thyroid cancer have been identified, we would detect thyroid cancer in approximately 5% of patients. So this is prevalence that we do see in general population as well. However, if there was a family where 3 or more members were affected at baseline, when we screened remaining non-affected family members, we were able to detect thyroid cancer in up to 23% of patients. So we think that this is truly genetic disease, while if you have 2 members affected, it still could be a by chance occurrence. What we learned from screening, that we are able to detect smaller sizes with lower rate of central neck lymph node metastasis, less extensive surgery therefore is required, and lower rate of radioactive iodine therapy as compared to those that were affected at enrollment. This is just because we were detecting it earlier. Obviously, we should not run into risk of overtreatment here. Standard of care should be implemented. So let me discuss about another clinical trials that are available in NCI, National Cancer Institute. So this is a clinical trial that is not only available at NCI, this is multi-center international basket trial. Basically, this is matching molecular signature of certain tumors, including thyroid cancer, with known medications that target particular mutations. So this is ongoing, many centers in the United States, many centers worldwide. Just FYI, National Cancer Institute investigators have such an option at the NIH to enroll patients and match their molecular signature with treatment. Clinical trial in pediatric thyroid cancer. And with that, I can certainly thank you and happy to take questions. Maybe let me stop sharing because I will be able to see those questions better. All right, chat box. I don't, I see just communication between the virtual host and moderator, but I do not see questions themselves.
Okay, let me read these to you. The first one, are there NIH studies specifically for people who have had a hemithyrectomy?
As a part of this natural history protocol that started in 1977, we do follow such patients over time with yearly thyroglobulin measurements and ultrasound. Usually these are low-risk patients, so usually we do follow them for 5 years, but this cohort could help addressing the question, what's the significance of thyroglobulin levels? Is yearly imaging truly necessary? So yes, not devoted clinical trial towards patients who underwent less than total thyroidectomy, but part of this bigger cohort of patients with thyroid nodules and thyroid cancer.
Thank you. Are you interested in stage 3 multifocal metastatic papillary cancer?
Yes, certainly yes. So, so again, this TNM staging system is associated with survival, right? While American Thyroid Association staging system tells us, addresses the question, what's my likelihood of persistent recurrent disease? Usually stage 3 corresponds to intermediate to high risk thyroid cancer, most likely local regional disease. So such patients may benefit from therapy with radioactive iodine, and we determine it based on both post-operative thyroglobulin levels as well as diagnostic whole body scans, trying to and other imaging modality if needed, trying to identify if there is uptake of radioactive iodine outside of the thyroid bed, suggesting potential persistent disease. If such scenario exists, we offer such patients therapy with radioactive iodine. If there are just lymph nodes, it could be empiric. If we observe distant metastasis, let's say in the lungs, we tend to offer dosimetry-based approach, so this individualized radioactive iodine implementation.
Thank you. I think you've already basically addressed the clinical trials for people who've had total thyroidectomies. Is that correct? You pretty much have addressed that.
Yes, so yeah, we do offer, if there's any additional treatment needed for such patients, we offer dosimetry-based radioactive iodine therapy for patients with radioactive iodine avid disease, redifferentiation for patients who are non-avid and but potential candidates for redifferentiation therapy followed by radioactive iodine, and screening for eligibility for other protocols. Yes. Mm-hmm.
Is there anybody in the room today, right now, who has a question? We have a question.
Okay, so my daughter has had she had the thyroid removed 3 years ago and lymph nodes, and had the RAI, and then it came back 3 years later. So do you still recommend that RAI be effective going forward? Because they are recommending it, and we're a little leery about whether the risks outweigh the rewards.
Right. So, in regards to, it's all a matter about what's the size, what's the growth velocity of these lymph nodes. Because, you know, if there are tiny, stable, less than 1 centimeter lymph nodes in the neck, you can even choose watchful waiting, not necessarily any intervention. Frankly speaking, for local regional disease, so disease in the neck, so lymph node metastasis in the neck, surgery, if the disease is robust enough, large enough, is the optimal management. So
Well,
what
she
we tend
had the
to offer
surgery.
such
She has had the
Sorry?
surgery. They removed the tumors the second time, and now they want to do RAI just in case there's anything microscopic.
As an adjuvant. And is thyroglobulin detectable right now?
No.
And so, you know, if there is microscopic disease, so meaning that's not detectable by the ultrasound, yeah, I would not feel strongly about necessitating radioactive iodine at the moment. I would rather watch such disease for a while. Also dependent on how anxious the patient is about, you know, active surveillance versus intervention. And it is not, it is one of the options. Again, always ask the question, the risk to benefit ratio. So what are the side effects of previous radioactive iodine dosing, if any? Is there a dry mouth already present due to salivary gland damage? What
Yes,
is the dental health? Yeah, so, you know, there are longitudinal data from Mayo Clinic on observing low volume microscopic disease in the neck over time. And for many of the patients, the disease tended to stay there and doing nothing, basically. The growth might be observed in younger populations, so I assume your daughter, she might be young, so this may require closer follow-up and intervention at some point. But right now, my understanding is that there was total thyroidectomy with neck dissection, followed by radioactive iodine, followed by redo surgery, nothing bulky at the moment. I think this is the great candidate for active surveillance, watchful waiting at the moment.
My name is Will. I'm calling about my wife and specifically curious on trials that are, so for papillary thyroid cancer with the BRAF mutation that are RAI refractory. So basically the radioactive iodine didn't work. What trials are in process now? There was one that was basically using the TKI to try to get it to differentiate and then try the radioactive iodine again, or you know, what are the different trials that are out there, or techniques out there for that case?
We call it redifferentiation, and I could speak. So there, I'm sure that there is still ongoing trial in Memorial Sloan Kettering utilizing those targeted therapy. Alan Ho had a great talk before on different methods of redifferentiation and ongoing as well as planned clinical trials on that. In regards to our NIH group, we do it under this I-124 PET-CT based dosimetry. So for patients with BRAF mutated tumors, we expose them to at least 1 month of BRAF inhibitors, so dabrafenib or vemurafenib. And we perform diagnostic I-124 PET-CT. And based on dosimetry estimates, we calculate the dose that would not exceed 200 rads to the bone marrow. So those that it's safe to the bone marrow if there is uptake in those metastatic sites. So, and again, I did have those information about the clinicaltrials.gov identifier, and I would be happy to share it with whatever way the organizers tell me is the most efficient and optimal. along with the flyers. We do have also some flyers, so I'm more than happy to share it.